Aloha, this is Roger Brewer with the Hawaii Department of Health. This is a recording of a presentation I gave on October 11th, 2022 in Nashville, Tennessee at the first meeting of the Association of Vapor Intrusion Professionals. And first, what a great group. There were around 200 people there from all over the U.S., a few people also joining in from Europe. The objective of this group is to write some short documents on best practices for vapor intrusion to try and clear up some of the problems we've been having in the past. The title of my presentation was Fake Data, Collateral Damage and the Need for Reform in the Vapor Intrusion Industry. What I wanted to go over during the presentation was just some lessons learned over the past 20 plus years now that I've personally been working on vapor intrusion issues. I start off my presentation with a pop quiz just to get some people thinking about some of the problems we'll be discussing later on. Let's say your objective was to estimate the average age of people in some audience. So you randomly select 50 people from the audience and you ask their age and you calculate an average. It's pretty simple. Then later on you discover that most, if not all, the people lied. So the quiz, the question is, what's the most appropriate statistical test to address potential error in the data and estimate a reliable average? So keep that in mind. We'll get back to this question at the end of the presentation. I got started in the vapor intrusion industry in the late 1990s when I took a job with California EPA, the San Francisco Bay Area Regional Water Board. This is the first guidance that we published in 2001 at application of risk-based screening levels and decision-making at sites with impacted soil and groundwater. Screening levels had been around for a while, but this was the first guidance, definitely in California and most of the US I'm aware of, where we actually published screening levels for vapor intrusion concerns. What was happening then was there's a huge property boom in the San Francisco Bay Area, as well as the U.S. in general. A lot of development and property transactions were underway. This new issue of vapor intrusion comes up, which most people hadn't been looking at the time. A lot of banks and property attorneys, they wanted environmental clearance letters before they would approve large loans and property transactions. When the San Francisco Bay Area, there were thousands of properties with detectable VOCs in the groundwater. I used to say, if you can find a site in the Bay Area where you don't find a few part per billion TC or PC or whatever, let us know. Otherwise, the trace levels everywhere. So it's really critical at this point. We had a good staff, but not that big, but we need to be able to screen out these low-risk sites so we could focus on high-risk sites. So you can see in the upper right-hand corner, PC vapor intrusion screening levels. These are the levels, final ones published in 2005. Pretty easy to set an indoor air screening level, say for PC and other VOCs. The toxicity factors are available. You just plug this into an indoor air screening level calculation. You can see we calculated 0.41 micrograms per cubic meter for PC and indoor air. Well, next, we need a subslab vapor concentration. So what concentration of vapors under a building could impact the indoor air above this action level, this screening level? Well, we talked to a lot of building engineers, people familiar with ventilation and such, and structural dynamics of buildings, and also with some of the modeling that happened to be going on at the time. It came up with about 1,000-fold attenuation factor for the San Francisco Bay Area. So that gives us our action level or screening level for PC directly under buildings. This is for residential occupancy of 410 micrograms per cubic meter. But we really needed a equivalent groundwater screening level for this, because that's what was happening in most of these sites, initially getting groundwater samples. So through modeling and a lot of field calibration where we looked at groundwater data and then soil gas data, then we came up with a groundwater screening level for PC, for vapor intrusion concerns, of 120 micrograms per liter. So now we had a tool we could really quickly screen out low-risk sites, and especially identify high-risk sites. One of the first sites we worked on in the Bay Area was a former chemical manufacturing facility. And at this facility, they'd been doing pump and treat and had a huge excavation and soil vapor extraction for years and years, I think at least 15 years. We were watching the SVE results, and over time they went asymptotic, so really not getting much out of the SVE system anymore. And our office issued a no further action letter for the responsible party. And meanwhile, they were constructing houses on this facility. The project manager for the site looked at the SVE data. This is right after we had published our screening levels, and realized that the concentration of PC and a lot of other VOCs in the stream of the SVE data was way above our action levels, over 100-fold. So it suggests a potential vapor intrusion concern. Well, we'd already issued an NFA for the site, our supervisor at the time. Again, vapor intrusion was a very controversial idea. You even need to worry about it back then. They said, we catch some and we miss some. Well, we were still concerned about this, so the project manager sent an email to the developer just to let them know that we had this new guidance out and it suggested potential vapor intrusion concerns at the site. And the developer, much to their credentials, I would say, realized the potential long-term liability of building the houses there, and they added vapor barriers under every single house. Didn't add a lot to the cost of the house, maybe $10, $15 a square foot when it was costing $200 a square foot to build houses. But a good move on their part. So we caught that one. We have similar high-risk problems with vapor intrusion elsewhere. Use some examples from outside the U.S. One is a school that was constructed on a similar site. And the kids inside the school, along with the teachers, started to get sick after a few weeks. And they realized it was vapors coming up through the floor of the building. I think it was chlorobenzene. They eventually had to close the entire school down. I'm not sure what happened to it after that. But now it's a site in China on the right. There's no messing around. This was a solvent site I helped work with. And here they dug up the entire plume down to about 50 feet, an area the size of a football field. There was existing slab at the site. They built a ex situ thermal treatment plant on top of the slab and a wastewater treatment plant on the facility. This whole facility is several square kilometers and treated all the soil on site. So yeah, not a lot of messing around now. And that's the key thing with the screening levels and such and vapor intrusion in general. You need to really rapidly identify high-risk sites so you can incorporate the cost of dealing with these problems into the redevelopment plans and budgets. In general, as long as the cost of cleanup is less than 10% of the overall redevelopment cost, then it can be managed. But one issue that several consultants in the U.S. have brought this up is you don't over-engineer the mitigation systems for vapor intrusion if you end up having to leave some contamination in place, which is fairly common. But doing this will help you avoid stalled construction projects, budget overruns and such, and avoid unforeseen legal and financial liability. That was pretty common news to the people in the audience at this conference, but something I think a lot of people hadn't thought about, at least in the EPA and other agencies, was that most investigated sites are inherently low vapor intrusion risk. So a lot of properties are tested just as part of a property transaction, or maybe the owners expanding their business or selling their business, or they're retiring or whatever. And the banks or property attorneys want to see some environmental data before they close the deal. So looking at all these sites in general, I'd say the high-risk sites, the ones that require a lot of cleanup, maybe 1 out of 500. That's probably being generous. It's probably less than that, where you have an expensive cleanup that's required at the site. Moderate risk sites, this in my experience, maybe 1 out of 100. These are all the sites where some type of sampling is being carried out. Most regulators don't see 75% of the sites in general that are ever tested because there's really not much there. But moderate risk, maybe 1 out of 100. You do some cleanup or some engineered controls. But the biggest concern we've been dealing with over the years are these really nothing small sites. There's never a lot of contamination there. It was low risk from the start, and it should be NFA'd, no further action from the start. To have trace subsurface VOCs, the owners, responsible parties typically have limited time and funds, property transaction, whatever is already in place. A site-specific assessment going into a lot of detail just isn't an option. This is where I get to my point of collateral damage. There's a lot of money that gets wasted on these nothing little sites just to get an NFA. And imagine if you're a small business owner, your retirement is basically tied up in this property, in this business. You're trying to sell it. And you have to waste $50,000 or $100,000, who knows what, just to get a letter from the state saying, oh, never mind, there's no big problem there. So that's a big issue. As one person told me years ago, I can either send my daughters to college or I can pay to clean up this little pocket of gasoline contaminated soil. You tell me what to do. We let them manage that in place. So you get delayed transactions, a big issue. Regulators get flooded with these nothing little sites, and they end up getting put on the low priority risk or list and just ignored. So the business owners or property owners are really screwed, for lack of a better word, being able to get their transactions done. And in a lot of cases, the properties just get abandoned because the regulators don't have time to look at it, and there's so much confusion over what's high risk and low risk. So that's what I focused on a lot in the presentation, how we can avoid these issues. As I used this slide in the past in other presentations, these health effects. We're always worried about risk and health effects. There's elevated cholesterol, elevated blood pressure, increased risk of stroke, increased inflammation, decreased immune function, decreased hormone function, mind fog. So is this TC? Well, no, you can look it up on Google. This is just typical health effects caused by stress. And that's, again, some of the collateral damage we have to deal with. So a lot of times as regulators, in theory, we're trying to help these people from poisoning themselves. So this is a pertinent quote here by C.S. Lewis. We have to be really careful what as regulators, we force private entities to do. So one of the topics that I mentioned, I didn't go into a lot of detail on any of the topics I'll be discussing in this presentation, but one was on petroleum vapor intrusion fallacies, something we've been dealing with for 30 years now. One of the big problems we see at a lot of sites is the site actually getting NFA'd, no further action by regulators, even though there's a lot of petroleum contamination there. And for vapor intrusion, the biggest cause of this is just to focus on BTEX and maybe naphthalene. And the consultant would be told by the regulators just to look at BTEX, naphthalene data. And if those are below screening levels, then no problem, no risk. Well, gasoline vapors as well as diesel vapors are overwhelmingly dominated by aliphatic compounds, not BTEX. And we did a detailed field study in 2012. We collected field samples of vapors at lots of sites. We looked at EPA's PVI database, and they all said the same thing. So even though the TPH aliphatics might be less toxic molecule for molecule than BTEX, just because of their overwhelming presence, they're going to drive risk. We've had toxicity factors for these groups, these carbon ranges of aliphatics and other aromatic compounds since the 1990s. If you have a toxicity factor, you can calculate a indoor air action level. You can see it for gasoline, 290 micrograms per cubic meter. It's in our guidance. Diesel fuel, it's got these lower length aliphatics plus some of the longer chain aliphatics. The longer chain aliphatics are a little bit more toxic, so the indoor air action level is actually lower, 130 micrograms per cubic meter. We have our attenuation factor. And these factors here reflect what we're using now in Hawaii, a 2,000-fold attenuation. So Subslab soil vapor screening level for gasoline, 590 milligrams, for per cubic meter. And then for diesel fuel middle distillates in general, 260 milligrams per cubic meter. So we can do this. We've been able to do it for 30 years, but it's surprising how it's still ignored in so many areas of the U.S. because it's not an EPA guidance. An EPA is essentially lobbied out of the petroleum business back in the 80s, so it's really up to the states to come up with their own guidance. Some of the fallacies we've been dealing with, first, middle distillate fuels like diesel, they are volatile. That's something I've always found odd, with some people claiming it's not, diesel fuel isn't volatile. We'll just go up to the gas station, go up to the diesel pump next time you're at a gas station and take a whiff from the end of the diesel pipe. And you can smell it. What you're smelling are all these aliphatic compounds. So it is volatile. And you can develop risk-based screening levels for TPH. We've never been able to do that since the 1990s. And then, so maybe you can, but then there'll still be a claim, well, BTEX, benzene would still drive risk, right? Well, no, if you look at the field data and compare action levels for the TPH to, say, benzene and other compounds, even when benzene gets below its screening level, your TPH aliphatics could be well above their screening level. We see that all the time. It's always the case for middle distillate sites, often the case for gasoline sites. We have presentations posted on our webinar on this. And then some people will make the claim, well, the aliphatics are going to degrade very fast in the subsurface, so we still can only need to worry about the aromatics. Well, it's the exact opposite of what we see in the field. The aromatics are often removed from soil vapors. Most of the sites I've ever looked at, a lot faster than the aliphatics. One thought is it's because the aromatics are so much more soluble, so they're absorbing into soil moisture. And then, okay, well, that's the case, and there's another case been made, well, the aliphatics are only going to get no more than 3 feet from the source or so before they degrade below levels of concern. That's not at all what we see in the field. still need a source separation distance. I would say at least 15 feet. I think we use 30 feet in our guidance. And in the end, you just remove the source to the degree practical for petroleum, and you manage the rest in place. That was the only thing we talked about for petroleum. Again, we have a lot of papers and recorded webinars posted to our webpage on the chemistry and toxicity of petroleum vapors, and a discussion of our TPH action levels, screening levels. Similar to California, there are screening levels. We helped develop those several years back. We have a TPH case studies that we posted when ITRC was preparing their TPH risk document. And this was a companion document to the ITRC document. So I worked with several consultants on that. And you can see the links down below for the references and recorded webinars. That was one topic. Then petroleum. And again, the biggest problem is regulators NFAing these petroleum sites and the developers going in to redevelop the property and start digging up all this stinky soil. And the project has to shut down. It causes huge headaches. So we need to look at this ahead of time. There's also a lot of discussion in other presentations about use of screening levels, conservative screening levels for initially screening sites for vapor intrusion risk. Already went over the ones that we prepared in California in 2001 initially, and updated up to 2005 and such. Again, these are mostly used to screen out low risk sites. It's a key point to get those people out from under the bureaucrats. And one thing you want to make sure you're doing when you prepare these screening levels is take into consideration cumulative risks. So maybe there's more than one compound, like BTEX. So looking at non-cancer risks that could be causing health concerns. So you never want to set screening levels to a target non-cancer hazard quotient, as you see up here, to 1, or very rarely, definitely not for individual compounds. We typically set it to 0.2, so there could be up to 5 compounds at their screening level. So 5 times 0.2 is 1. As long as you're below a non-cancer hazard index, cumulative index of 1, then you're okay. And you can use the screening levels too to confirm high-risk sources before you start testing indoor air. One thing when you're doing this indoor air samples we'll talk about later, you always want to focus on the source VOCs, not other ones. So these are the screening levels for initially screening for vapor intrusion risk. There's also a lot of discussion about rational risk goals for cleaning up sites, and this is modified from some guidance that Tennessee just published. I think they were collaborating with New Jersey on this. So the screening levels are set very conservative. In this case, you see here less than a 10 to the minus 5th cancer risk, or 1 hazard quotient or hazard index cumulative. So the screening levels are set where mitigation isn't going to be required. When you get between a cancer risk of 10 to the minus 5th, 10 to the minus 4th, or a cumulative non-cancer hazard index of 1 to 3, that's where you might need to initiate some passive or even active remediation. And definitely when you start exceeding a cumulative cancer risk of greater than 10 to the minus 4th, 1 in 10,000, or a hazard quotient, a hazard index of 3 or greater, you definitely require some active remediation, plus or minus some passive. So a lot of people agreed on this in the conference. We didn't spend a lot of time on it. What we spent a lot of time on in the presentation and in a lot of side meetings was the way samples are collected in the environmental industry versus the commodities industry. It's the same objective in each one of the industries. You're trying to estimate the mean concentration of whatever you're looking for, either commodity or contaminant in some media. There's a whole field of science that's virtually unknown in the environmental industry until recently, just referred to as the theory of sampling. It's a standard for the commodities industry, but it's a new idea for the environmental industry. So in the commodities industry, you can be looking at, say, mining, where you're collecting samples from a large stockpile of crushed up ore, again, to estimate, say, the concentration or ultimately the mass of gold in that stockpile. Agriculture, you may be testing for viruses or other problems in stockpiles of grain. Recycling, testing for rare earth metals, elements in recycled, say, catalytic converters. Same thing in pharmaceutical industry. You need to test and ensure that the mass of some drug in a pill meets the quality standards. How do you know there's 200 milligrams of X drug in this pill that you're taking? And same thing in the oil and gas industry, we're doing oil and gas exploration. So in these types of commodities industries here, they have correct use of what's referred to as infinite element sampling methods. Infinite element just means you can't test individual particles. So you collect a single large sample. It's collected from multiple points within a designated decision unit volume of the targeted media. And we'll talk about this again in a minute for vapor intrusion issues. That's very common. And these are referred to as multi-increment type samples. In the environmental industry, the only group that's properly collecting samples is the indoor air groups. And here they don't refer to it as this, but they're using infinite element sampling methods. Again, they're going to collect a single large sample from multiple points within the indoor air, say as the air is circulating through. We'll look at this in a minute, in a designated decision unit volume of the indoor air, say over a certain time period. The other main types of environmental sampling for soil, for groundwater, and soil vapor use the incorrect use of what's referred to as finite element sampling methods, or discrete and grab samples. This is a holdover from survey sampling or testing of populations, where you can test individual elements, individual people. They carried on the same idea for testing of soil and groundwater, but we can't test individual particles of soil or individual molecules of water or soil vapor. And this whole approach of collecting single small volume discrete samples and then independently analyzing every single sample isn't reliable at all for the environmental industry. We talk about this in a lot of our other presentations. That's why in the environmental industry, we spend a lot of time just kind of chasing our tails. There are a lot of books out on theory of sampling. They're just completely overlooked, again, in the environmental industry. So you see a collection here on the left by various internationally known authors. And some on the right we'll look at in a minute. So just a few of hundreds of example references. If you just want to get a general idea of the theory of sampling for these infinite element type media, then these two guidances published by the Association of American Fee Control Officials, really useful. They talk about decision units and representative samples and such. And in this case, it was discussing it or presenting it for testing livestock feed or food crops, but it really applies to any media. We published a two-part paper in 2017. We did a field review of discrete sample reliability. And that's posted on our webpage along with recorded webinars. Kim Espensen out of Denmark is one of the world experts in the theory of sampling. He just published a book about 2 years ago. And then at the very bottom here, the paper we published for a conference in Norway. It's the World Conference on Sampling and Blending. Most of them were miners, but you had a lot of people from agricultural industry and pharmaceutical and recycling there, and a few of us from environmental industry. You can see it's a similar title. Again, we have a lot of field studies and sampling guidance, recorded webinars on this subject. In this presentation, I just wanted to go over the basics for the group at the Vapor Intrusion Conference. And so here's some just basic ideas of how to collect a representative sample. We see that all the time in guidance and regulations, but there's no guidance on what that actually means. So whenever you're working on a project, there's 4 simple questions you want to start off with, basic theory of sampling concepts. First is, what's the question? What is the investigation question? We'll look at some examples of this in a minute. What's the decision unit? What's the volume of media that you're concerned about? And how can a representative sample be collected from that decision unit? And then ultimately, what's the decision statement? So what are you going to do when you get the data? You don't want to get the data and then think about what to do. You want to have some kind of decisions up front before you go out and collect a sample. One thing to keep in mind as far as investigation questions, there's only 2 reasons ever to collect environmental samples. One is to assess risk. The second is to optimize remediation. This includes isolating source areas. Next, let's apply these concepts to testing of indoor air and subslab vapors, either to assess risk or to optimize remediation. So first, we'll look at indoor air sampling to assess risk. This is already well done. They don't really explain it in terms of theory of sampling, so I'll be discussing, but That's essentially what everyone's been doing for the last many years. First, what's our question? And our question might be, does the mean concentration of X in DU volume of air Y exceed the screening level or risk of Z? So the first thing to think about is what's our decision unit. So the decision unit is the volume of air circulating within the occupied or the area of the structure or the area of the structure of interest during the specified exposure period. So for example, default home volume in a lot of vapor intrusion guidance is 244 cubic meters. If we assume a 24-hour exposure period and an indoor air exchange rate of 0.35 times per hour, that's the minimum indoor air exchange rate required for permits for building homes, then our decision unit volume is over 2 million liters. That's how many liters of air are circulating through this home in 24 hours. That's our DU volume. So what's our data need? Well, we need the mean concentration of X in this indoor air during the 24-hour period in this DU volume under normal operating conditions. So here we're sampling to assess risk. I know in some guidance documents in some states, they require that people turn their HVAC system or their ventilation system off before sampling, and I've never understood this. It doesn't make any sense with respect to the question being asked to assess risk or not. So we definitely wouldn't recommend it. So we'd collect a sample under normal operating conditions. Well, how do you collect a representative sample? Ideally, you'd send the entire volume of circulating air to laboratory. So this beach ball I have in the figure here is pretty much to scale the volume of air circulating through that house in one day. That's obviously way too big to send to a laboratory for testing. So the worst thing you could do, which I don't know of anyone that would do this, indoor air folks anyway, is to collect a small volume, say 1 liter instantaneous sample, where you go in the house and open up a Suma canister for 10 seconds or whatever, 1 liter, and close it and send that to lab. Well, they would know that's not likely to be representative of what's the air circulating through the house. So using a theory of sampling approach, which is what they do, you'd collect a single continuous sample of circulating air during the targeted exposure period. So you'd put a Suma canister, for example, or you could use passive samplers out. And as the air is circulating through the building, then the Suma canister will be set to continuously collect a sample during your exposure period. Now, keep in mind the concentration of the contaminant or the VOC in any individual small volume of air is irrelevant with respect to the question being asked. We're always dealing with chronic exposure risk. We're looking at indoor air or at most exposure to any environmental media. The screening levels that are published are based on exposure over several years. So I know there's some cases, say for TCE, there's some studies done, or a study done many years ago that suggested subchronic risk or risk posed by exposure to TCE over a few hours or something. I know there's a lot of controversy in the study from what we've discussed with other toxicologists. We don't think it's very reliable. We mostly focus on chronic long-term risk. But either way, you're still not looking at any individual breath of air, trying to test that in the house. You're still looking at exposure over probably several days, if not several weeks or several months. So a large volume of air. That's done pretty well. And indoor air sampling to optimize remediation, that's also done pretty well. And here the question might be, where and what is the primary source of indoor VOCs? So here typically what would happen is you split your structure in the house or whatever up into individual rooms, individual areas. So you're looking at spatial heterogeneity. This tends to be especially important, trying to isolate the source so you can remediate it. You can also, in some cases, use continuous monitoring where you're taking small samples of the air in one fixed location every few minutes. So you're looking at any temporal heterogeneity, trying to figure out what's going on in that particular room that's causing the VOC concentrations to spike high at certain time periods. But one thing to keep in mind with continuous monitoring is that the question is never what is the concentration of X in any given breath of air, so it has no pertinence to risk. Again, at minimum, you're looking at several days probably of exposure, if not years. And it's not risk-based. It can help you figure out the source, but the concentrations reported in continuous monitoring really can't be directly compared to risk-based screening levels. So first, in this case, what's your decision unit? What's the volume of air circulating within the targeted room during the specified exposure period? So what are our data needs? Again, it's the mean concentration of X in indoor air for the specific room during the specified exposure period. So you're splitting the house up into smaller and smaller decision units in order to isolate the source. So how would you collect a representative sample of that? Well, ideally, you'd send the entire volume of air circulating through that part of the structure to the laboratory for testing. Again, that's not practical at all. Worst thing you could do, which I don't think anyone would do, any of the indoor air folks, is just grab an instantaneous 1 liter sample, collect over a few seconds, send that to the lab. That's not going to be reliably representative of the air circulating in the room over the exposure period. Using theory of sampling approach, which again is what's generally done, you collect a single continuous sample of air circulating in that specific part of the building during the exposure period, exposure time. So I joked during the presentation that Aristotle was thinking about vapor intrusion way back when. And that's a key part, is understanding what resolution you need to make a decision about any type of environmental risk. And typically you start off with one decision unit for the entire structure. You start breaking it up into smaller and smaller areas and maybe smaller and smaller time periods until everyone's satisfied that you'll be able to assess risk as well as optimize remediation. We talk about this in other presentations on theory of sampling. That was indoor air. How about subslab vapors? Well, it's the same approach, the same questions. So first, how would you sample to assess risk? And I would say this is mostly very poorly done, except for the few exceptions. So your question again is very similar. Does the mean concentration of X and the DU volume of subslab vapor, Y, exceed the screening level for risk Z? So the first thing you look at, again, is what's your decision unit? Well, this is the volume of subslab vapors anticipated to intrude a structure during a specified exposure period. And typically we're looking at exposure periods over many years. Well, that's not very practical to test vapors coming up over many years. By the time you finish testing, the risk has already been incurred. So the default vapor entry rate for intrusion into houses, a lot of vapor intrusion guidance is 5 liters per minute. We talked to a lot of building engineers. They think that's reasonable of leakage through the floor. So 5 liters per minute, expand that over, say, 24 hours. We just want to look at the vapors intruding over 24 hours. That comes out to about 7,200 liters. That's USEPA's default volume of intruding vapors, just expressed in terms of 24 hours. Again, I've got the beach ball there roughly to scale, and the weather balloon there you see on the right is roughly equal to 7,000 or so liters. So what's the decision unit? It's the volume of subslab vapors anticipated to intrude the structure during the specified exposure period. In this case, let's just focus on 24 hours. I mean, again, that's way too short. That's what we can do practically. What's the data needs? What is the mean concentration of X? We want one number in the intruding vapors during the target vapor intrusion period. How would you collect a representative sample of that? Again, just like we talked about for indoor air, you find out where the vapors are intruding, or maybe you have some hypothetical vapor intrusion point, and you collect 7,000 or more liters of air out from underneath the slab and send the entire volume to the laboratory for testing. The worst thing you could do, as was the case in indoor air, is grab to assess risk, is to test some small volume, say 1 liter instantaneous sample of vapors under a slab. What's the chance that's going to be reliably representative of vapors intruding over a 24-hour period? Pretty low to nil. Under a theory of sampling approach, you'd collect a single continuous sample of vapors intruding a point in the slab over your specified time period. And again, the concentration of the VOC, whatever you're looking at, any individual small volume of intruding vapors is irrelevant with respect to the question being asked. Well, how do you do that? Here's actually a guy, Todd McAllery, I'll show you the reference later, with Geosyntek's been doing this for more than 20 years. So sampling to assess vapor intrusion risk. If you look at our guidance document, you see the references there, and I'll show you those later. We refer to these as large volume purge LVP subslab vapor samples. In this case, what you're doing is you're mimicking the investigation question in the design of the sample collection method. So you see here in the photo, and this is a field study we carried out, that the building's gone, but the slab was still there. This is a dry cleaner site. We already knew there were some vapors under the slab. So in this case, not really sure where the source is, so we put a vapor test point in the middle of the slab, and then you run PVC pipe down to that point, hook it up to a shop vac if you're testing for chlorinated solvents, an air compressor with a guard air type sampler if you're looking at petroleum vapors, just due to explosion concerns. And you turn it on, and then you, as it's purging your target volume, say 7,200 liters, then there are flow meters there, and the woman in this picture is monitoring the flow. We have a Suma canister that's tapping into this PVC pipe, and it's collecting a continuous sample as the vapors are being purged out from under the slab. So now the sample we send to the laboratory is representative of 7,200 liters instead of just some instantaneous volume. So a much more useful type of sample data we're collecting and much more directly representative of the question that's being asked. In this case, we wanted to test for over a 5-day, hypothetical 5-day vapor intrusion period. So we actually purged 5 times 7,200 liters, over 35,000 liters, and collected continuous samples from each one of those purges. Here's the data from that study. This is published. It's in our guidance and report on our webpage, 2017. So on the left, LVP results. This is PCE. You can see sample 1, 2, 3, 4, 5. We could have just done one sample, but we wanted to split it up to look at how the vapor concentration change. Starts off at 17,000 micrograms per cubic meter. The last sample we purged, 54,000 micrograms per cubic meter. This is something you'll see most of the time when you're doing these type of samples, purging. Large volume purge is the concentration is going to increase with the more you purge, because you're starting to capture these isolated hot spots under the slab that you would have totally missed with your single discrete sample. So you're not diluting the sample at all by increasing the purge. In collecting a sample, you're increasing the representativeness, and chances are you're going to be increasing the concentration. That's what we did in this study, and it's posted to our web page. The average concentration after these 5 purges, 41,600 micrograms per cubic meter, way above our commercial industrial action level. Again, these LVP sample data, they're routinely going to be higher than what you're going to predict with these small volume, discrete subslab vapor samples. So you miss a lot with these small volume crab samples under slabs. Not that surprising to people who've done a lot of this. Some of the references, see Todd McAllery, 2010 document. He called it high purge volume. We call it large volume purge sampling. Same idea. All we did in our guidance, in our study, we just attached a risk-based volume to how much vapor you need to purge. That's really the only new thing we did with respect to what Todd was doing. We're not that concerned about where the vapors are coming from under the floor at this point either. We're just assessing risk. Todd spends a lot of time looking at vacuum changes over different parts of the slab to see where the vapors are coming from. Don't really need to be worried about that at the point we're just assessing risk. Then we have webinars, detailed posts published to our webpage on this. That was subslab vapor sampling to assess risk. Now let's look at subslab vapor sampling to optimize remediation. And this is kind of mixed results, I would say, and we discussed this in a paper we published in 2014. So the question in this case, again, similar to indoor air, is where are the highest areas of subslab vapors? You're just looking at where the highest vapor source is. So the traditional approach with these discrete small volume grab samples under the slabs, you get really pretty maps like the one you see here. It's after Mort Smith, some paper he published in 2013. But the problem with these, and you can see on the far left, if you test around any individual point, then you're going to see a lot of random variability of the concentration of the VOC just around a single point. So in the case on the left, the concentration of PCE, and these are passive samplers, but they did a really good job estimating the actual concentrations. So the range 28 micrograms to 61 micrograms, just within 2 feet of each other. What if your screening level is 50? And these, you can actually convert these pretty well into concentrations. Let's say your screening level is 50. Well, depending on what sample you collected around that point, it might show up as a hot spot or a cold spot. And that's the problem. We talk about this in other webinars we give, especially in our field study of discrete sampling reliability for soils. You get these false hot spots and cold spots in these isoconcentration maps. They're just artifacts of random small-scale heterogeneity. Move your point over another foot or two, and you can get a totally different number. So your cold spot can become a hot spot, and your hot spot can become a cold spot. So you have to really be aware of these types of maps. Anytime you see isolated hot spots and cold spots, they could be totally artificial. In this particular example that Mort Smith did, this particular site, you see the big orange spot in the bottom center. That was actually real because he knew that there was they'd been dumping PCE solvent in that spot. But in other cases, these could be totally false. So again, what's the decision unit here? Back to these 4 steps. Decision unit is the volume of subslab vapors within any targeted area. So you'd split this slab up into different areas. And then your data needs, you need the mean concentration of X in targeted area of the subslab vapors. What's the ideal way you would collect representative samples? Where you split the slab up into different areas and you send the entire volume of vapors underneath that part of the slab to the laboratory. Well, that's not going to happen for a lot of reasons, just getting it out. And you're going to be disturbing the plume as you collect the sample. Worst thing you could do, again, maybe it's the best you can do for now, but it's still not very good, is test these individual single small volume instantaneous samples. So when you do this, the large scale map patterns, they might be real, but again, you have to really focus on these small scale hot spots and cold spots and just assume they could be false, but large scale patterns might be real. Under a theory of sampling approach, which should be developed for this, then you would still designate DU areas, smaller areas within the slab, and collect multi-increment type subslab vapor samples. Let's look at how to do that. Well, first, actually, just getting back to the artificial hotspots. This is a part of a presentation we gave on our field study of the random variability of discrete soil samples. This is actual field data here. This is a site we knew it was contaminated with lead. We set out 24 grid points, and each grid point we collected lots of individual discrete samples. We could essentially estimate a range of concentrations of lead in soil in a discrete sample that you might collect around any one of these grid points. So example, this grid point number 21 here, and any discrete sample you collect around that grid point, you could get a concentration of lead of 103 to 419 milligrams per kilogram. So think about that. It's totally random, the number you get. If your screening level is 400 or 200, any discrete sample there, it could be below, it could be above, totally random. So here's just a hypothetical example. Somebody goes out to the site, each one of these discrete points, 24 points, collects a discrete sample. And here's a map they make. Let's say the next person comes by, they go to the same points, collect a sample, and look, they get a totally different map. The hot spots start jumping around because the sample they collected at the point had a higher or lower concentration. So this is just showing how these hot spots can be totally artificial. You can keep doing it. Here's another 2 people go out and they collect samples, same points, just a few inches apart. We're even testing the same jar of dirt, a different pinch of soil, you get a totally different number. And here's your iso concentration map. Keep doing it, and you get your iso concentration maps, your hot spots just bounce all over the place. This shows you how artificial these iso concentration maps can be. We don't realize that because we only collect one set of samples. Typically you do. At this site, we didn't. Just a word of how do you address this problem? We're again back to theory of sampling. This is in the study we did in 2017, the report we published at high density subslab passive sampler investigation. So in this case, the same slab where I was showing you the LVP sampling before, we split the slab up into 25 different grid cells. It's 8,000 square feet total. In each one of the cells, we know there's a lot of variability in PCE concentration of the slab, and really just based on how much money we had, we put in 4 passive samplers in each one of the cells. And then so we installed 100 passive samplers, but then with each cell, the 4 samplers at the laboratory, these were done by Beacon Environmental, they combined the carbon strips from each one of those 4 passive samplers into one test. So only analyzed 1 sample. So 100 samplers installed, but only did 25 analysis. So by combining a 4 increment subslab vapor sample in each cell, then we were able to better capture some of the variability within there. It wasn't ideal, but it's definitely an improvement. And you can see the map we produced on the right. The blue is cold, the red is hot, yellow is in between for PCE on the slab. You see where we collected the LVP samples. I showed you the 5 series of PCE data right in the center of the slab. Remember, the concentrations were going up. But it turns out, we didn't know this at the time we collected it, there's a diesel pipeline under the slab that used to feed a generator at this dry cleaner. So what it looks like happened is that as we continued to purge from that LVP point, and it finally reached into the hot area under where the diesel generator was and where there was an underground tank there. It started drawing in some of the fumes from that area. I guess it was a PCE tank in that area. Also, there was an earlier study done when they did 12 standard traditional 1 liter grab samples in the slab, and you see in the upper left of the second figure that one hot spot on one sample. It was completely wrong over the vapors that were there, and that's still something that baffles us. But you see the one hot spot, that throws up a red flag anyway. It just shows you how unreliable these traditional subslab vapor samples can be. So that was it on the ideas on testing for risk and testing to optimize remediation. Indoor air already done pretty well. Subslab vapors definitely could stand some improvement. I think these LVP type samples to assess risk, or maybe some type of multi-increment subslab vapor samples to try to isolate higher areas of the vapor plume would be useful. Let's get back to the pop quiz. Here's pop quiz number 2. See, the objective is estimate the average concentration of contaminant X for some set of samples that you send to the laboratory. Let's see all these subslab vapor samples that the lab tested for us. So your consultant collects, in this case, with indoor air samples, in this example from hundreds of homes, uses the data to calculate an average concentration of background VOCs in homes. So we use this example. You later discover that laboratory equipment wasn't operating properly during the analysis of most, if not all, the samples. It's a similar question. What's the most appropriate statistical test to address potential error and estimate an average for that data? Well, what do you think? There's nothing. You can't do anything with that data. The database in general isn't useful. None of the individual data points are reliable. So there's no statistical test that can take care of unreliable data. You just have to toss your database and start over. So it's the same question as the very beginning. We're asking the average age of people in an audience, and it turns out all the people in the audience, or most of them, or a lot of them, lied. You can't really do anything with that. That's one thing when you collect the data yourself. And I always say the same thing for my stocks. I bought them; they must be really good. Even though they start going down, you realize you made a big mistake, and it's really hard to let go and admit that you made a mistake. This is called the endowment effect in psychology. And that's a big issue for a lot of the work we do with vapor intrusion in the environmental industry in general. Now, this leads straight into the EPA's empirical database for vapor intrusion. I know a lot of people have been using, but think about it. First, if you look in the upper left here, in this case for the database, they estimated subslab attenuation factors by first they collected a sample of indoor air in hundreds of homes, one sample from each home. And that was probably good data. Probably 24-hour sample at least. But then they collected the instantaneous, these small volume subslab samples directly under the slab of the building. And then that ratio they defined as the subslab attenuation factor. It's the concentration of indoor air divided by the concentration of subslab vapor. Well, that would only work if, as they say here in California's 2011 guidance they published, that would only work if the subslab vapor plume is reasonably homogeneous. Well, subslab vapor plumes are never homogeneous, and I've never seen a single study that would suggest that. So if you look at under Law et al. In 2009, it's just a petroleum plume. They were mapping out this TPH data. Depending on where you collect your sample under that slab, you get a totally different attenuation factor. So it's a completely invalid approach to do it. It sounded good at first. It was useful exercise to do, but it doesn't work. So you've got that issue of sort of large scale variability, and even around individual sampling points, move it over a foot or two, you get a different number, and it could be totally random. So the big issue with this, people have been pointing it out since before 2010. They started the database in early 2000s that your database isn't valid, but it was published by the EPA folks anyway, which caused a lot of angst. So the error in the data is not quantifiable. You can't fix this again with some statistical analysis. There's some folks would say, well, it's conservative because it comes up with a really low attenuation factor, only 30-fold attenuation. Well, it's not conservative at all. It's scientifically invalid. It would only be conservative if a a valid, scientifically valid approach was used to come up with it, these attenuation factors, which they aren't. So that's a huge mistake in the vapor intrusion industry, thinking that this default subslab attenuation factor is conservative. So it's not. It's fake data and it's in all its glory. And a big issue is this erroneously flags an overwhelming number of these low-risk vapor intrusion sites or VOC sites we talked about before. It causes a lot of collateral damage unnecessarily to these small businesses and property owners. So we're still trying to get that this document retracted. The EPA published in 2012 and again in 2015. Don't wait for that to happen, folks, especially the state regulators. EPA is not going to retract guidance that they've published in the past. They never have and they never will, even though it shows to be erroneous. And we can go into the psychology and the issues behind that, but that's just a fact. So a lot of people, and you can read the paper we publish, understand this. It's not that complicated, but really need to quit waiting for EPA to solve our problems. They have a lot of great people in EPA, good technical support, but we have more in terms of vapor intrusion within the states now to work with. As an alternative way to estimate subslab attenuation factors is to look at the estimated vapor entry rate for, say, a typical home in the U.S. divided by the indoor exchange rate for that home. This is done in a lot of detail by groups that look at building ventilation and energy efficiency, ASHRAE among others. We published a paper in 2014, split the U.S. up into similar climate-based zones as they did, and then based on the average number of days that the homes are heated versus cooled and a default gaps, size of gaps in the floor, we can calculate an average vapor entry rate for each one of these regions. We also have data on the typical indoor exchange rates for residents in these regions. So we can come up with a much more reliable and accurate subslab attenuation factor. I don't have time to go in a lot of detail with this here, but you can see the paper we published below. Also, there's recorded webinars posted to our webpage. We go over this in detail. Just for example, a typical subslab attenuation factor, again, based on the building leakage studies by these groups, been doing it since 1940s. For cold regions, might be about a 300-fold attenuation, 0.003. As you get to more warmer regions, your attenuation factor is going to increase because the homes are heated less. So you have a lower rate of vapor entry over much of the year until you get out to the tropical regions like Hawaii and southern Florida, where you have a estimated subslab attenuation factors to default of at least 1 to 2,000. So again, when you look at it this way, and based on a much larger database, you come up with more realistic attenuation factors based on what people are seeing in the field. So for more details on that, you can see the reference below. That was a fairly quick overview then of different problems that we're seeing with vapor intrusion studies in the U.S. and ways to get around it. So just to review some points, you always want to consider TPH for petroleum vapor intrusion. We want to publish reasonably conservative but still useful risk-based screening levels, again, to quickly screen out these low-risk sites and get these small, nothing sites out from under the bureaucracy. Then utilize rational target health risks for requirement of active remediation. Also, for assessing risk, we're looking at subslab vapors, and it really should be using large volume purge type sampling methods so we get data for a much bigger sample of vapors. It's based more on risk, the question being asked. Consider some type of multi-increment sampling methods to isolate vapor sources under slabs, optimize remediation. You can do this especially with passive type samplers. Also, we talked about quite a bit in the conference, don't require indoor air testing until you've identified a high-risk subsurface source. And this imposes a lot of financial and burdens and other problems with the initial investigation of sites where there's no indication of significant vapor intrusion risk. And also, you're going to keep your guidance evergreen so you can incorporate new research and field experience. That was the presentation. I went into a lot more details here than I did in the actual conference in Nashville. We use this slide at the end. I've used this in other presentation just to show there's still a lot to learn and to improve in our vapor intrusion investigations. It's a graph of confidence versus knowledge in field. I'm not sure who did this. Someone sent it to me. That's a big problem we have. I think in anything we do, not just vapor intrusion, all aspects of life. You initially think you know everything about something. We start to realize maybe we don't quite get it, or some people are getting something totally different than we are. You have to get through this, we call cognitive dissonance hurdle, go through your 5 stages of grief. There's more to this than I thought. Maybe I'm never going to understand this. Probably where I was with vapor intrusion 15 years ago. And that's starting to make sense, and then kind of the aha moment. So we need to move through this in the vapor intrusion field. Now, I'll end with this quote from Jules Verne: "Science has made mistakes, but they are mistakes which it is useful to make because they lead little to little by the truth." Thanks.